Thank you very much. Thanks very much for inviting me to give this presentation about the biobank. I was just one of a small team to start with the biobank, and during this presentation I'm going to tell you a little bit more about our team and what we have been doing. So to move here, I just press the...
Sit down, you've got the pointer.
Good, got it. I have the... Thank you, I'll manage. Right, I'm overwhelmed by the quality of the presentations, and also thank you very much, Stephen, for your optimism, because I think the moment is right for ME CFS, as you say, and it's an opportunity that needs to be taken. On the other hand, we're all too familiar with a situation where funding is very scarce for this particular area of research, and we heard from Alan today, from Eliane and others, about how little money has come, or how little amount of funds have come to this field. So it's not difficult to imagine a situation where you as a researcher, perhaps a young researcher, you have a hypothesis which relates to a pathophysiological mechanism that could explain some aspects of ME CFS. So what you do, you formulate a hypothesis, you did that, and to test this hypothesis, let's say you decided you would like to investigate the presence of a particular marker in the blood in people with ME CFS, and if this marker is increased, or if this marker is present, perhaps you are giving a very good contribution to the field. So what you do, you plan your study, and you think about who are going to be your cases, who are going to be your controls. Perhaps you want healthy controls. Perhaps you want also sick controls, people who have a fatiguing illness, like chronic pain, for example, with fatigue, that will serve also as controls. So you're going to decide how you're going to select your patients, how you're going to obtain information in the best possible way. You're going to think about the blood collection, where you're going to store until you have your sample size. Let's say you calculated you need 200 cases and a similar number of controls. Sorry to be a bit lengthy on that, but this is slightly on purpose, because I just want to show you or remind you, as for most researchers, unless you have direct access to samples, that's what you go through. It's a lengthy process, and it consumes lots of resources, because you want to make sure you have the right participants in your research, the right samples and the right data. And that's where biobank comes into it. And this is not my presentation. Down. Good. So that's why one of the reasons why biobanks have been becoming more and more popular recently is because biobank, of course, is a repository that stores and manages biological samples for use in research. So there are three key components there I want to highlight here. You talk about stored biological samples linked to high quality data, and which are collected with the objective of doing research, and in particular with the objective of sharing with the community. And so if you think about collaboration, the biobanks is a type of infrastructure that facilitates that. And why you would like to use biobanks? Because you get access to samples and data who have been standardized and quality assured, so you are likely to get high quality samples and data. And of course, as I just mentioned, this will be very cost effective and time effective ways of doing your research. How much does it cost to do research using a biobank as compared to not using a biobank but going through all the process is something that is difficult to say, because each research is different. But you can find some literature around which you say that you might be saving 90% of your resources by the time you start actually analyzing your samples. Of course, this will depend on the type of study that you're doing. And then you can use the biobank, of course, for a number of hypothesis testing, understanding disease pathway, investigating disease subgroups, and biomarker discovery. That's the sort of sort of things or research priorities, if you like, that you can benefit from biobank and are very relevant for ME/CFS in other areas. So I'm going to tell you a little bit about what the UK ME/CFS Biobank offers as a resource, and particularly having in mind our sharing objective. And then I'll just tell you a little bit about our journey or how you got here, and then I'll tell you a bit more about the research that we do at London School. So what we have as participants, we have nearly 600 participants now. Of course, some of them, about half of them, I would say, have at least one follow-up with 6 to 12 months afterwards. Some of them have monthly follow-ups for a time period, and an increasing number of them, of these all participants, are having now their third follow-up visit. And in all follow-ups, we collect blood samples, and we are starting to, and in some cases, other types of samples as well. And of course, data, there is a data collection at every encounter. The plan is in 2 years we'll have some patients at least, some participants will have 5 follow-ups. So all cases are confirmed medically. They receive a medical diagnosis, and after the medical diagnosis we revisit their clinical symptoms and we classify them according to Canadian consensus criteria, CDC 94 and Institute of Medicine criteria. And of course, we consider other diagnostic criteria as well. Some people may meet other diagnostic criteria. Our population is age 18 to 60 years, and we include from the milder forms of ME/CFS to the most severely affected, and these include those who are pretty much bedbound or housebound. So we have our nurse visiting patients at home. Our controls include healthy controls and a sick control, which consists of people with multiple sclerosis. Right, so what is available in the biobank? We have blood products, and this includes for each participant and for each time point, and this includes whole blood, serum, plasma, red blood cells, PBMCs, and blood for RNA. So we have a diversity of blood derivatives that are available. And at the moment, we have just over 30,000 aliquots stored at the biobank. If you haven't shared or used them for our own research, we would have more than that. Every single participant provides an average of 46 aliquots of different products. So it's very important. Samples by themselves are not enough if you don't have good quality data attached to them in most instances. So we try to clinically phenotype them in a way that is useful for research. We see all patients or participants face to face. We get their blood pressures measured, seated and standing, and now we're moving to repeat blood pressure measurements over 10 minutes period. So that's the type of the diagnostic test that was used for Nina, for example. So that's something that we do. We have the hand grip strength in all individuals, and we found so far very, very interesting profiles of hand grip strength in our cases, particularly the severely affected. And we have other clinical tests. We are piloting now pain, quantitative measurement of pain. We are still deciding whether we're going to be brave enough to offer to our patients. Results of routine lab tests, I think I mentioned, this is important to mention because all our recruits, they go through a panel of blood tests that are useful to exclude other conditions and also for further phenotyping. And then you have other data. Of course, we'll have social demographic data. We have comprehensive data on the disease itself and how it appeared, triggers, etc. And in particular, we applied CureME symptoms assessment questionnaire, which is a questionnaire that enables us to confirm whether cases which have already received a clinical diagnosis actually meet one of the study criteria, which means generally Canadian, and the Fukuda and IOM criteria. The IOM, of course, we started halfway through because it was not available when it started the biobank. Then we have the UKMEB. UKMEB actually stands for UKME CFS Biobank, is just the abbreviation for short. We have the Participant Phenotyping Questionnaire, which is a lengthier questionnaire than the first one, and it enables us to attribute severity of different symptoms. I'll go back to this in a moment. And then we look at quality of life. We use SF-36, for example, pain and fatigue scales, and general health questionnaire for mental health. So just give you an idea what types of data we have available. As I more than once already said, is an open resource for academic and commercial users. And of course, we have some priority areas which are in line with our priority areas for one research at the London School. But basically, what I would say that anybody with an interest in biomedical research into ME/CFS can apply for samples if they have a good hypothesis that would be beneficial to be tested using the samples that we store. The procedures are in the in that link there. I don't know if you can see from the back. Apologies for not, but this you can just Google as the UKME CFS Biobank and you find instructions to apply. But basically, you submit your application in an online form to start with to our steering committee. And our steering committee has 3 members that rotate and they judge initially the applications. And one of these members are our chair, who is actually Dr. Charles Shefford from the NE Association. One of our members, the other member is a patient representative, and the other one is a researcher. We submit these applications for peer review, and then of course we have ethical approval to distribute the samples, but we require every applicant to have obtained their own ethical approval. And at the end, the scientific subcommittee at University College Hospital, Royal Free Hospital Biobank, where we actually store physically our samples, need to say the yes. So it seems like a lengthy process, but I think it's very important to ensure the ethics behind the sharing of samples. And I think at the end of the day, it's less burdensome than it seems. In a couple of months, we usually have the samples ready for distribution. And then, of course, all of them, the aliquots distributed are de-identified. So sometimes I have the impression that it's not very easy to see from the back. I don't know if it's too oh, but you can see there. That's excellent. I don't know, can I, basically, how are we doing in terms of sharing samples? So is this black, the red one is the, oh yeah. So, so far we distributed, the green ones and the yellow ones mean different things. The greens, we already send the samples. The yellow, people can be in the part of the application process. We're discussing that, but we haven't actually sent the samples. So we distributed to Brazil. We have United States, North America. We have some places in Europe that we send samples for, and we have even in the Middle East, but it's in Israel actually, it's not in Syria, because Israel is a tiny country just beside Syria. We're not getting samples from there. And the UK, we have Oxford and Cambridge universities as well, UCL. We are discussing one interested person in Cardiff and actually 2 in Scotland at the moment. Right, so so far I mainly told you about the Biobank as a resource for sharing, but let me tell you how you got here. It has been a 5 or 6 year journey, so it's really hard work. And we're pleased to have reached this point. And of course, there's lots more that we can do and plan to do. But all started about from our conversations with people with ME. So all started by trying to find out from people with ME what they wanted to facilitate, to energize research in ME/CFS. And therefore the idea of the biobank is as much an idea from people with ME as I would claim is ours idea, because it's a project that started from the first moment in partnership with the ME community. So we had some focus groups with people with ME, CFS, and then we had some workshops as well with researchers, with clinicians, with expertise in the field, and also in particular, and this is very important if you want to set up a biobank, we discussed our ideas with experts in human tissue banks, law, and ethics. So I'm not going to go to the qualitative studies we did with patients, but I just want to illustrate with 2 slides what people had told us as part of these qualitative studies. And this is one quote from one participant with ME, and they say it's about ensuring quality and linking to relevant data. And they say, this person said, if it's done like really thoroughly, so the samples are going to be very good quality and have the appropriate information attached to it. Because if you don't know what area it's going to go down, we have to be quite broad in the information. So that's something that's what we're trying to do, actually. That we heard from someone who participated in our discussions and qualitative studies. Actually, we didn't do this biobank for a particular study, because this often happens. You do a particular studies and then you have some leftovers and then you can keep for other studies. No, we want to have a biobank which will serve the research community. So we need to think about different possibilities, different hypotheses, and how this will be fulfilled even in the future. So we try to be as comprehensive as possible in terms of type of data and samples that we collect. And then there is another saying here, which is very important, because people have their concerns. People with ME are worried sometimes about, in this case, they said they are afraid what you're going to do with the samples, whether the government would get hold of it, whether insurance companies could use it, or whether benefit agencies use it. We can understand where they come from. And we need to be very careful, because when you have the samples and the data from people who generously gave for research, we need to respect the ethical principles and respect and be grateful for this gift that they give, and treat the samples and data in the best possible way. And of course, at the same time, you want to have the most benefits from the biobank. So of course it's very important to balance the dangers of misuse that we cannot afford to have and the potential benefits that we want to maximize. And this just illustrates the ethical procedures that you need to go through in the beginning of our project. So of course we need to have IRIS we need to have MREC, IRIS. We need to go to research and development offices in different parts of the country. We have a London School Ethics Committee approval. The London School has a Human Tissue Authority license, otherwise you wouldn't be able to have a biobank. And also the Royal Free, where the samples are physically stored, has, of course, Human Tissue Authority license. Very important is the informed consents and free withdrawal. Everyone donating samples and data to the biobank can withdraw. We have one withdrawal so far. And of course all samples are identified, so we make sure, if we can, that nobody will be identified through any mechanism during any research that is done. Of course, there will be brilliant hackers out there that can go everywhere, but I think we are very careful with the confidentiality. Not going into the consent process, just to remember, informed consent means informed consent, and we give plenty of opportunity for people to actually clarify what they are committing to, and also they are always reassured, as I said, with the opportunity to change their minds. So that was the process. A lot of conversation with experts, a lot of conversation with people with ME, considerations of technically active terms, and we ended up with a protocol. And then we started to collect samples and data, and which are stored at a Royal Free Hospital biobank. And this actually is an important thing because we have a disease-specific biobank, which is for ME/CFS, and for MS to some extent because they are sick controls. But they are stored in a general biobank, which is the Royal Free Hospital biobank in London. And the importance of this is much more cost-effective to do this way, and also enhances the quality. Ensures the quality of how the sample's been stored. Of course, they are 24/7 attended, the biobank, so they are alarmed, and they have all the mechanisms and all the business plan to ensure, to ensure the right procedures are in place in case of any incidents like lack of energy, etc. So it's very safe, it's very secure. In the same way, our database it was developed specifically for the biobank, and of course it respects all the criteria for a safe database. Right. So I told you a story which is quite long. We're talking about 5-6 years, and that's where we are. And as we said, we have longitudinal data. We have nearly 600 participants. But I would still say this is a good beginning because this is the journey that needs to go much further. And one of the things we have been discussing a lot as part of the CureME that Eliana presented earlier is and Eliana mentioned this very briefly, is the synchronization of the way data is collected, the type of data is collected, how cases are diagnosed for research purposes, and how samples are collected across Europe. So the red dots represent in Europe the places where there are existing bioresources. And of course, there are countries, as Eliana said, where very little is known about ME, and they are not still in a position to have a biobank. But all this is a process, and maybe one day there will be more dots here. And also we have been discussing a lot with North America about synchronization, harmonization of biosamples. Right, so that's CureME team. So the biobank itself is mainly Eliana and Caroline. You met Eliana, many of you will know Caroline. So there are those people who actually are looking after the biobank most of the time. But we have a larger group than that, and of course we are researchers and we're clinicians, some of us. And I just want to point out within our team that these are the clinical researchers, if you like. Our main strength is in clinical epidemiological research, but then we have what we call a lab-based research team. And Hazel Dockrell, Professor Hazel Dockrell, is a professor of immunology who works with Jackie Cliff. You're going to hear from her tomorrow on immunology, gene expression, and a few other interesting things. And Professor Tani Clark is our genetic epidemiologist who is looks after statistics and our genomic studies. So I'm not going to be saying too much about our research today, but you have the opportunity to hear Jackie with a few examples of our research, exciting examples of our research tomorrow. And also, I was very pleased to see the presentation before mine about the socioeconomic implications of ME/CFS. And this illustrates that it's not only samples that we share, we also share data. And this has been useful in other projects as well. So let me tell you about the research at the London School. So that's the main areas of research that we have expertise. As I said, clinical and epidemiology, immunology and infection, genomics and gene expression. And then, of course, we have an interest in health education and disability and socioeconomic assessment. So it's quite a broad areas of expertise. And when I talk about direction of travel here, I'm actually talking about our strategy that we created about 5 years, 6 years ago when we started, and we keep updating that. But I think it's very pleasing to see that this is very it's very similar to what I'm hearing from you, that should be the direction of travel. So this convergence or perception of what is needed in research in ME, I think is something extremely important to realize. And this transcends the UK. We can see similar thoughts in Canada and of course in the US and Europe. So it's about, I think, genetic environmental predisposing factors. This is an area that it's very important to pursue forward. It's very nice to see the presentation on genetics of chronic pain and some interrelationship with fatigue. Clinical and molecular biomarkers, that's from different omics. We heard a lot about that already today, not least from Mark and of course from Alan. And then better diagnosis and stratification of cases. Certainly it's not about one, it's not about 4 MEs, it's about different diseases which will have overlapping mechanisms and distinct mechanisms, and we need to get to the bottom of that. And I think as a research community you're probably going now in that direction. And at the end of the day what you want is better care and treatment for ME/CFS. I think the ultimate goal is that people who have ME/CFS and their families and their friends will feel that something is done for them and it will be helping them. Just a few. Am I okay with time?
Yeah, okay.
Go. I'll just show you a few of our data, and this is not, as I said, is not very in-depth analysis, but it's more here, and I think I have 4 slides on that, just to give you an idea what type of data we have, and what type of data is available for researchers. For example, this is the impact of ME on quality of life. So what we see here is our healthy controls, and this is the different domains and summaries. Those are the 2 summaries I think we call, and those are the 8 domains of the SF36. I gather most of you, many of you will be familiar. And normal people, these are normalized scores. Normal people will have for all domains, normal in inverted commas. I don't think there are. I mean, I try to see those without any fatigue and illness, but I'm not sure what normal means. And there are not many around, I think. Not here in the world. So body pain, road, so they are pretty much as expected. It's between 50 and 60 in normalized scores. Then you go to MS, and you see the green one is MS. And of course MS as a neurological disease is quite disabling. And then we have ME, which is much more disabling. And you see in all aspects. And what I would like to point out is the mental component summary, which is a summary of the mental disability, if you like. It's not very different from ME and MS, but when you go to the physical function, in spite of MS being a physical disease, and I don't like to use this terminology, but I recognize to give physical disability, the disability in ME is actually worse. And you can go through in detail with the different domains and do, but I think this is something that reinforces how disabling ME/CFS is. I mentioned we have some longitudinal data, and this is about, there was a question here earlier today about outcomes. I think it came from Jeannie, if I'm not wrong. And fatigue is not the best outcome. And neither is physical component summary or mental component summary. But then what is the best outcome? I think we're still trying to reach that point when you have better outcomes. But until then, I think there are outcomes which are useful to measure improvement. And if I take, for example, the physical component summary, this graph shows the following. About 50% of people with ME/CFS at baseline, 6 to 12 months later, they will have about the same level of physical disability based on this particular indicator here, which means that the score that they get is not significantly different from the score they got at baseline. So we know it's a chronic illness, and not unexpectedly, most people, at least half of the people, will not show any change in their disability level within 6 to 12 months. But about just over 25% will have some improvement. Maybe not all of these are very clinically significantly, but some of these will be clinical significant improvements. And we can look at how those people are and how they're different from the others. At the same time, unfortunately, some people get a bit worse. But in most cases, this is very subtle. But we're talking about 6 to 12 months. So hopefully we'll be in a position to look in a couple of years for what happens over 5 years, for example. This is one way to aggregate symptoms. We aggregate our symptoms, and this is one way of phenotyping, trying to subgroup in a very, very crude way. And I'm just illustrating by immune-related clusters. So we take some indicators, mainly from questionnaire, which would indicate immunity dysfunction, if you like. And when you look here, it's difficult to see, but this is healthy controls. These are MS and these are ME. And in this case, 100 is the maximum, and this is the average. And so if someone has 80, like here, for this particular symptom, then they have quite severe disease. So this is an indicator of severity of different symptoms, which are aggregated into immune-related clusters, autonomic-related clusters. Look at the difference between ME and CFS. ME is worse than MS in terms of severity for both, except for bladder dysfunction. Surprise, well, bladder dysfunction is one of the MS symptoms that, not for everyone, for some people, and then we have a few others which are quite similar between ME cases and controls. And this is the neurocognitive clusters. Again, healthy controls, pretty okay here, nearly 0. Then we have MS, and then we have ME. So in different clusters of symptoms, and we have 7 in total, we have the severity of ME symptoms tend to be worse than MS. And this is just one slide that relates to that. We look at the 7 different cluster of symptoms in terms of severity. And we look at again, the physical component is summary from the SF-36. This is the summary for physical disability. The main predictor in MS of physical disability, I'm oversimplifying the way I'm phrasing this, but basically that's what it means, is neurocognitive symptoms. For ME, in mild cases, are the post-exertional symptoms. And for the severe cases, are neuroendocrine symptoms. And for mental component summary, for multiple sclerosis are the autonomic symptoms. Then for ME are the sleep dysfunction. This is what making people feel mentally disabled, is sleep dysfunction. But you have ME severe is neuroendocrine, both for physical and for mental component summary of the SF-36. What does this tell us? It's a very crude analysis here, but there are lots of quite strong correlations here, the p-values you can see here. It shows that mild and severe cases, the symptoms that bother them most or limited them most are actually different from one subgroup to the other. Right, so I'll just summarize before you finish. So thank you very much for listening and hearing about what I had to say to the UK ME Biobank. I think partnership was at the center of everything from the beginning, and until now we have, as I mentioned earlier, our steering committee is heavily represented by people with ME, and this is what makes our steering group so powerful, I think. We believe we have a robust protocol, cost-effective design. The sharing data and samples are something that we are in a position now to do, and we have been doing for the last year or two. And I invite all researchers here who would like to discuss about their needs for data and samples. We're very happy to discuss, of course. And of course, at the same time, those who have bioresources and they have their ways of collecting data and samples will be very happy also to dialogue with this with you, because maybe there are ways we can improve the way we collect data and samples, and maybe vice versa, other people can also improve in some aspects of their collections. But the objective of that would be that at the end of the day, if you have collections like this one and other collections here in the UK and across the globe, which in a way have some synchronization, some harmonization, this will allow large studies to be conducted with samples we have similar data and similar way of procedures for collecting samples and similar samples, and also it will be very useful for replication studies, for geographic comparisons, and a number of other things. So I think this would be very key, and I really look forward to discussing more with different people, and I think collaboration is the key here of this meeting. So I think this gives a wider opportunity of collaboration, and of course it allows our CureME team at London School to do multidisciplinary research in ME/CFS. And I just put in this at the very end because we don't have a biobank because we wanted to do research. Of course we want to do research. Of course we are funded mainly from the NIH in the United States to do research, but the biobank itself we created for us, but especially for other researchers and for the patient community. ME Association and other charities have supported it, and these are some of our partners who help in different ways in our data collection and sample collection and in other ways. Thank you so much.
Sure.